First off, I would like to say hello once again to everyone in the course and thanks in advance for taking off the time to watch my presentation in the absence of my presence. Unfortunately, I do not have access to the projector at this time, so I will record myself during this presentation for you all to have someone to read it for you. I hope it doesn't bore you all completely. My topic for this presentation was the relationship between methogens, methanotropes, with the root system of marine plants. Therefore, I decided on the article titled Methogenesis and Methanotropy within a Sphagnum peatland. I will first start this presentation off to mention methane because it's probably the most important aspect involved in the process. Methane, as we know it, CH4, is a colorless, odorless gas that occurs abundantly in nature. As we all know, methane is lighter than water and slightly soluble in water. It is known as a product of anaerobic bacteria decomposition of vegetable matter underwater, which does not require oxygen for its growth. It also burns very well in air, therefore forms carbon dioxide and water vapor. Emissions of methane are important because methane is radioactively 20 times as active as carbon dioxide in the atmosphere. This makes it difficult to separate net methane emissions into gross production and internal consumption. Based on field results, maximum methane consumption occurs at 6 to 12 centimeters depth. The pH, light, oxygen, and relative moisture content have significant influences on methane consumption within wetlands. These factors vary depending on the active microbial community, because without this there would be a decrease in oxygen and temperature. Studies have shown that zonation in the distribution of methogenesis and methane consumption. They have not yet demonstrated this through microorganisms, methane oxidizing, and methane consumption within the wetlands. Methane's chemical structures are known as a tetrahedral molecule because all of its four substituents have the same angle. Just a quick view of the different ways to look at the chemical in a different perspective. Enough of methane. Okay. The northern peatlands are located in Concord, Massachusetts. Their water tables are characteristic of low nutrient levels and low pHs, normally around 4.0, which would be acidic in nature. Therefore, the deposits of peats can accumulate due to decomposition occurring because without the acidic water tables, this phenomenon would not occur. With the water tables being below the surface of the peats, there will be an unsaturated zone of depth. This is inhibited by anaerobic bacteria, which we know has no oxygen. Because of the anaerobic bacteria, carbon is released as methane, which diffuses and bubbles into the atmosphere. Unfortunately, some gets consumed by oxidation of carbon dioxide by methanotropic bacteria in the unsaturated regions. The total peatland methane emissions are therefore dependent on the activities of both methogenic and methanotropic bacteria. In order for methanogenic and methanotropic bacteria to be in existence, the peatlands had to be in certain conditions. And without these methane productions of methogenesis and methanotropy, there would not occur. If there is an increase in the oxygen and temperature, the microorganisms in the peatlands will increase. As mentioned earlier, this study was conducted in the northern peatlands in Concord, Massachusetts. These peat samples were obtained from Burroughs Bog located in Concord, Massachusetts. The methane consumption in the samples were established and its location of activity were also determined. Phospholipid fatty acids, also known as PLFA profiles, were obtained to confirm the presence of methanotropic bacteria. Therefore, the determination conducted methanotropic bacteria within the microbial community. To test these samples, oligonucleotide probes for 16S rRNA were used to quantify the methogenic bacteria. The peat samples were taken from Thurles Bog in Concord, Massachusetts, in an acidic bog, which covers about 0.4 hectare in the Merrimack River watershed. The vegetation is composed primarily of Spignium species and small shrubs. The water tables are 7 to 14 centimeters below the surface of the peat. Precipitation is a must because it provides the only hydrologic input to the bog. On the other hand, evaporation accounts for about 80% of its output. The temperatures range from 32 degrees Celsius to 22 degrees Celsius in the month of August. These times were from noon to night time on the days that were sampled. Therefore, the peats had a 2 degrees Celsius to 9 degrees Celsius increase throughout the day. The Spignium species is briefly mentioned in the article, so I will elaborate on it. I had to look it up because I was unsure about what it was, and the article has limited information about the Spignium. The Spignium species consists of about 100 to 130 species in the world that are associated with habitat environments like bogs, fens, and swamps. The Thurles Bog it imitates a bowl-shaped floating bog that is carpeted with Spignium mosses that are geographically isolated from the habitats. This is the actual scene of a Spignium moss from the Thurles Bog in Concord, Massachusetts. The samples were collected in a stainless steel cylinder with the dimensions of 12.5 centimeters internally, and bandsaw blades were welded to its bottom for support. For the methane production experiments, plastic cups were used and placed over the bottoms to minimize the contact with the atmosphere. Therefore, the intact cores were transported to the laboratory for analysis. Peat profiles were used in order to determine nitrogen fixation. Acetylene, which is a known methanotropic inhibitor, which will be used for nitrogen fixation. 4.0 gram samples were placed in 120 ml serum bottles sealed tightly with air. Nitrogen was also added to 0.1% by volume to each treatment. The control set had no nitrogen added, as this was to validate nitrogen fixation with 2.7 micromole of methane. Another set was incubated under 200 microeinstein of light. The last test was incubated for 48 hours at 22 degrees Celsius. These samples were then removed from bottles and dried at 70 degrees Celsius for 20 hours. Afterwards, the samples were grounded with mortar and pestle and screened through a 0.5 millimeter mesh and combusted at 1000 degrees Celsius in a Hershey CN analyzer. Lastly, the Finnegan CT box cryogenically separated the products and the nitrogen. The bacteria were first isolated from the peat samples in order to isolate the nucleic acids. These peat samples were blended with homogenized solutions and then centrifuged to remove any of the soil debris. The nucleic acid preparation had an A260/280 ratio greater than 1.5 blotting and hybridization. For the measurements of PLFA, peat samples collected on 2 days and fractioned into 0 to 2.5 centimeters and 5 to 7.5 centimeter fraction. Then the samples were lyophilized with 1 gram aliquot extracted and analyzed for PLFA content. Cores placed in anaerobic glove boxes with a N2H2 atmosphere. Within the glove boxes, the serum stoppers were utilized to contain methane, in which there was no methane loss during the experiment. The headspace sampling took place for 110 hours and were determined using a Schmidt-Steuer GC80 gas chromatograph. Subsections of peat were divided into 1.5 to 2.0 grams, placed in 120 ml serum bottles, and then filled. Aqueous concentrations assumed to be 3.64% of headspace concentrations based on Henry laws that have a constant of 661. These concentrations determined at 10 to 20 hour intervals for 98 hours. Lastly, there were 7 methane concentrations that ranged from 15 nanomole to 15 micromole. Methane consumption present in the top 9 to 11 centimeters of peat were absent below 11 centimeter depth. The maximum methane usually occurred at 4 to 6 centimeters. And this was uniform at around 4 to 6 centimeter range. As you can see, I have no picture here, but figure 1 was unable to post. If possible, the article is attached for you all to view, and you can have a clear understanding of what I have in this slide. Methane production activity samples had depths of 5 to 10 centimeters and 30 to 35 centimeters, and the methane produced outside this range was not detected. There is evidence of an influence by temperature to the rate of reduction from 10 degrees Celsius to 22 degrees Celsius. Also, oxygen concentration had influences in rate and extent of methane consumption. See figure 2 on the section of article, which I was unable to post. If at all possible, the article is attached for you all to view. Methylogenic bacteria have strong hybridization of 1.25, 15, and 20 centimeters in May. There were clear peaks around 25 to 30 centimeters, which were consistent with methane production rates. PLFA analysis provided evidence for the existence of methanotropic bacteria. The existence of microorganisms in the superficial and methanotropic layers are 5 to 7.5 centimeters depth. The population is composed of gram-positive organisms, gram-negative obligate anaerobes, acetomycetes, anaerobes, methanotropes in the deeper layers. Their microbial abundance is greater in the superficial layers. See Table 1 on the section of the article. Because I was unable to post, if at all possible, the article is attached for you to view. Nitrogen-fixing organisms were present throughout the peat core. Nitrogen that was greater in the upper layers of the core. Nitrogen was undetected light, or methane. This took place in 0 to 4 centimeter sections and were not detectable in any other fraction down to depths of 20 centimeters. See Table 2 on section of article because I wasn't able to post it. If at all possible, the article is attached for you all to view. Methane consumption rates increases relative to methane concentration. Methane consumption should be significant to temperatures as low as 10 degrees Celsius. There was no activity observed at 4 degrees Celsius. Temperature influence is dependent on original depth and peak of the peak samples. Methane consumption rates peak at temperatures increasing with a decreasing depth. See Figure 5 on this section of the article because I wasn't able to post. If at all possible, the article is attached for you all to view. Discussion. Combining methane consumption and production assays with the molecular analysis facilitated both localization of methanotropic and methanogenic bacteria and a determination of the environmental factors which influence them. The cores examined had high methane consumption rates of 4 to 9 centimeters below its peak surface. The presence of methanogen bacteria, as determined by PLFA analysis, agrees with the reports of Thurlsbaugh and 2 other locations that were examined. There were high levels of methane consumption in the highest levels, while lower levels were undetected, possibly due to an insufficient oxygen level. Therefore, when the water table is higher, it will be relative to the surface of the peat mat. The methanogen population decreases below 30 to 35 centimeters, while the PLFA showed significant decreases between the top and the bottom levels. In conclusion, nitrogen fixation to Thurlsbaugh shows no significant influence on methane or light. Therefore, methane oxidizing bacteria or phototropic bacteria are dominant nitrogen-fixing organisms in aerobic peats. And I put this all in here for you all, kind of like a discussion to kind of put everything in perspective to show, to see if you actually can understand it, because it was a lot in the paper, so I kind of summarized it the best I could. So I hope this and discussion helped you to try to understand it a bit more. As you see, here are the references listed below. And if you'll go to the I think it's the third hyperlink. This is where you'll find this article. I got it from Elsevier off of jsu.edu. Okay, and that'll be the end of my presentation today. So thanks for your time and patience.